Welcome to this biochemistry lecture. We're going to today about biosignaling. This one's actually a little bit longer than what we normally have been doing. That's the reason I kind of cut back on carbohydrates a little bit. This topic kind of is quite deep, and it's in 460 and 461. This is another one of those topics that gets chopped off a little bit at the expense of other material, and I really feel like this is a super important topic for the pharmacist. And I'll tell you that, primarily because biosignaling is, in essence, how a large number of drug targets are targeted, I guess. These drugs interact with these receptors to block biosignaling, and so this is a super important topic that I don't feel like we do justice in some of the other courses. So let's go ahead and bring up a case study. 68-year-old man Delbert, who had sustained previous inferior and anterior myocardial infarctions, presented with gradually increasing fatigue, edema, despite increasing diuretic therapy. What would be your diagnosis? And you can pause it here and do some background if you'd like. The answer is congestive heart failure. And congestive heart failure occurs when the heart cannot pump enough blood to the rest of the body. And this can happen for multiple reasons, but basically you end up with a failure to pump blood that can sustain the body. So symptoms: you get nausea, fatigue, dizziness. It can be caused by a heart attack, heart valve disease, congenital condition. You can have failure to receive blood to the heart, which is coronary artery disease. You can have damage from toxins or illnesses, all kinds of stuff. And we have treatments that range from bypass to valve replacement to ventricular assist device. And the most relevant to you guys is we have tons of medications that can assist in this. This will be a major area for you to study, and we'll talk about how this mechanism works, or some of the mechanisms work to regulate this. You diagnose it by irregular heartbeat, so you're going to get an EKG. You can have swelling of the liver, leg swelling. You can also do other tests, cardiac stress tests again, which is MRI of the heart. Heart failure is a second leading cause of death. Actually, it might be the leading cause of death. I need to look at this. 6 million people in the United States are affected by this. You have a 1 in 5 lifetime risk after the age of 40. There are all kinds of risks, as very much like diabetes. In fact, they're all like diabetes. Decreased activity, obesity, poor diet, high sodium, high cholesterol diet, smoking, all those kind of things. And this is a nice little chart right here that shows the treatment options that are associated with congestive heart failure based on the problem. So if you have a class 1 coronary heart disease, you can get into lifestyle changes and some drugs, and then as you get higher, you get into transplants and ventricular heart disease. So we're going to talk, and there's different drugs, right? Beta blockers, ACE inhibitors, inotropes, diuretics. We're going to talk about angiotensin II receptors, blockers, which enhance the function by widening the blood vessels and reducing blood pressure. So that's what we're going to talk about today. And associated, the angiotensin II receptor is a GPCR, and I am going to I want to pound on this as pharmacists, because GPCRs represents the major drug target. It represents a major drug target. 30% of drugs, some estimates up as high as 50% of drugs, target GPCRs. And the hard part about GPCRs is there's a couple of hard parts that we haven't that exist. First off, they're incredibly hard to work with because they're transmembrane proteins, so you have to work with them generally inside a membrane, or you have to find a way of making them happy. Second off, and this comes from experience, we've tried to do several studies on GPCRs, and I've received several grants on GPCRs where we're trying to characterize them and work with them, and they're ridiculously difficult. The other aspect is, so there's about 6,000 known drug targets. Actually, a very large majority, or a very large number, to a majority are GPCRs. So when we talk about enzymes being 47% of drug targets, you realize that actually, in all actuality, many of those enzymes are also associated with GPCRs, and those enzymes are also important drug targets, or they're related to GPCRs. So all of these things you can see, of all the drug targets, DNA, integrins, we're going to talk about most of them today. So integrins, ion channels, transporters, we've been talking about them for the past couple days. This today is very, very important for for pharmacists and for drug development and discovery. Alright, so let's talk about signal transduction. Cells receive signals from the environment beyond the plasma membrane, so out in the world, from antigens, hormones, neurotransmitters, light, touch, pheromones. And these signals cause changes in the cell composition and function. Earlier we talked about membranes and how they give a brief overview of this process, and these can cause changes in the cell's composition and function. So again, when we receive a signal from the membrane, there are 5 properties, or from outside the membrane, there are 5 properties that occur as we take that signal, and that signal is brought into the cell and then acted upon. So first and foremost, it's very specific. And so the signal molecule fits at the binding site tightly. Other signals can't get in. We'll talk about that more in a minute. The signal gets amplified. So we receive the signal, enzyme 1 will amplify it a certain percentage, and then it goes to enzyme 2, which amplifies it again, it goes to enzyme 3. There's a cascade pathway that occurs. And related to that is modularity, where we take these signal pathways, and proteins with multivalent affinities can form complex signal with interchangeable parts. And so, for one signal pathway can use enzyme 1, 2, and 3. The next one can use 2, 3, and 4. Another one can use 1, 3, and 4. There's modularity, and these are that creates the specificity. The receptor needs to get reset, so there's a desensitization and adaptation set where the signal gets received, the signal gets sent down the line, and then that whole system has to reset, which is desensitization. And then last but not least, we have integration, and that occurs when we can have, like, let's just take estrogen. We can have multiple estrogen receptors that give a very unique profile to get a very highly specific response. And so we have 2 signals. Also, you have 2 signals that can have the opposite effect, and that can regulate the outcome. So we have multiple ways of doing this. Alright, so receptors specificity, let's talk about that. Some examples of ligands are small ions, things like iron. We have organic molecules, things like adrenaline, epinephrine. We have polysaccharides, things like heparin, fibroblastic growth factor, peptides, insulin receptor. You'll notice that here's what the small molecules on the left, colon, then we have the receptor. Proteins, we can have vascular endothelial growth factor, also called VEGF, and you have VEGF receptor. So we have they're very specific to their receptors is the important part of that discussion. Again, a molecule, one can come in and bind, but something else, even very similar molecules, cannot bind. The signals do not fit. And if they don't fit, you're not going to get a response. Again, this is another example. This is a protein-protein interaction where a protein can will come in and fit exactly, and it'll have an interacting residue. This is a SH2 domain where it binds a protein for phosphotyrosines. You can see the tyrosine right there, and when we say phospho, we're going to add a phosphate group to it as a post-translational modification. So this is an interaction of a PTB domain with a tyrosine residue. And you can look this up if you want to on the PDB at 1-SHC. And anyway, the moral of this story is very specific. You can see that red fits right in that binding groove of this partner protein, very tightly and specifically. Next, signal amplification. This is an epinephrine receptor, very similar to acetylcholine receptor, one epinephrine molecule, and this is why when you give a epinephrine shot for something like anaphylactic shock, you can see we start with 1 molecule of epinephrine. At the receptor, we amplify it. We get 10 times. When it gets from ATP to cyclic AMP, we are amplifying it by 200 times. We go from phospho-PKA, a kinase, we amplify it another 100 times. Phosphorylase B kinase, second kinase reaction, we amplify it 1,000 times. Another third phosphorylase B, 10,000 times. Glycogen to glucose, 100,000 times. When we get to blood glucose level, we're now putting into blood 100,000 molecules based on 1 epinephrine receptor binding series. So the important part is from 1 molecule we can amplify some of these signals hundreds of thousands, if not more. So signal amplification is very important in the process. We also have modularity. This gives an example of how we can use phospholipase C and or IP3, which are 2 intracellular second messengers. They produce the same or similar responses. So, if we have a hormone receptor rebinding, the second step is we get a GTP. This is a GPCR, so we get a G protein coupled response. Phospholipase C is activated. Phospholipase C then comes and interacts with IP3. IP3 binds specifically to a calcium-gated ion channel to release calcium. That calcium channel then interfaces with a protein kinase C, and that phosphorylation produces a cellular response to the hormone. You can see we have different modular elements. Sometimes they're also very similar, sometimes they're not similar, but all of those responses act in a pattern to give the signal. And that as well as amplifying the signal, it also provides an important step-by-step regulation, advanced regulation of the system. So modularity plays an important part in biosignaling. Again, this kind of gives so if we have each one of these represents a protein interaction, we can go down and have different responses to each signal that occurs. So we start stacking up these signals. So we go, so if we have for transcription, we can take, if we have a binding target is a proline-rich or membrane lipid PIP3, we're going to put those in series, and that specificity. But you'll notice that a lot of them overlap, are very similar. So these series becomes important. That provides specificity for the reaction. So another important component of this system of biosignaling is self-inactivation or desensitization. So when G proteins in the first 3 steps go through and they're sensitized and they complete their signaling mechanism, the GTP is dephosphorylated, and it comes back and resets the GPCR. So once the GPCR binds, in the second step, the hormone receptor causes a displacement. So once the G protein displaces from the GPCR, the hormone kicks out, and then a new GDP gets recycled and puts back into the system to reset the system. That's a very, very important part of the whole process, is resetting the system to allow the system to resend that signal. So one of the things that we study commonly is nonspecific binding of a receptor. Hydrophobic ligands can be a problem, hydrophobic in nonspecific interactions. And so when we do this in the lab, we kind of try to identify how nonspecific binding occurs in these receptors and in these enzymes. That's the problem. So receptor binding, if we're going to measure total binding, we're going to take specific binding and non-specific binding, we're add them up, and that's going to give us total binding. So total hormone added is L. That's the ligand. We're going to try to measure the ligand receptor interaction. So total hormone is equal to the ligand concentration plus the receptor and ligand interaction. And what we see here. Alright, so now that we know the general principles of these, we're going to go into specifics about the 6 general types of signals and how they can occur through the membrane. We'll talk about GPCRs, tyrosine kinases, guanylyl cyclases, which are they're a receptor class. We also have gated ion channels. We also have adhesion receptors, and we have nuclear receptors, which are common hormone receptors. So we'll talk about all different kinds here and how they work. This is a great visual representation of how these things work. So you can have all these signals can lead to gene expression, or they can come back up and open gated ion channels. So the main one the main class, the one we'll spend the most time on today, is GPCRs. And GPCRs, again, are alpha helical 7 alpha helical integral membrane proteins. They're heterotrimeric, meaning that there are G proteins are GPCRs have 7 alpha helix that have a form of transmembrane domains that allow for ligand binding. G proteins are on the cytoplasmic side there. G proteins are heterotrimeric alpha, beta, gamma. They're membrane associated. They bind GDP and GTP. G proteins mediate signal from GPCRs to other protein targets, so they're part of the signal cascade. We'll talk about that in great detail. So this is the general structure of a G protein, and there's numerous examples here, and you might note that they all look the same. The one on the left is a beta-2 adrenergic receptor. We have a ligand binding pocket that comes in. You can see what ligand binds on the outside of the membrane. On the inside we have the GPCR, or G protein, excuse me, with the alpha, beta, and gamma subunits. All of these have that. They abstracted that out. The G protein will bind to all of those. They've abstracted this out just to kind of to save some space. But you'll notice things like morphine analog, so it's an opioid receptor. We have histamine receptors, and in D you have a light receptor from rhodopsin. So all of them have very similar structures with 7 there's different kinks in the helices to provide different binding pockets and receptors. Some have prosthetic groups as D does, but generally they have the same mechanisms. Again, light, calcium, small ions, odorants such as pheromones, small molecules, amino acids, nucleotides, all these things can interact with this. Protein interactions are also mediated by this. And you'll note that the G protein, or the GPCR, is attached to a G protein. The G protein has an alpha, beta, and gamma subunits. GTP is translocated, and that affects some other kind of effector. From there we have intracellular secondary messenger that will cause some sort of an event, gene expression, opening of an ion channel, etc. If this B is the top view, and I think this is very interesting, these 7 alpha helices that cross the membrane are oriented in a way so that they can be the ligand can bind efficiently to provide specificity for that GPCR. Again, you'll note that these alpha helices sometimes are kinked, as you see in 6 here. Sometimes they're straight. Sometimes they're oriented in different ways. A lot of times they have an extracellular domain and an intracellular part to the same alpha helices. That's a pretty common way of putting these things together. The example we're going to talk about is epinephrine. We'll also talk about acetylcholine receptor as a GPCR a little later. Again, epinephrine is a hormone that's made in the adrenal glands. It's used to mediate the stress response, and it basically provides energy to the cell. And it provides it in 3 places. In muscle and liver cells, it induces the breakdown of glycogen. When in adipose cells, it induces lipid hydrolysis, and in heart muscles, it increases your heart rate. So how does this work? So epinephrine here, as you see this small molecule here, you can see has a lot of functional groups. It binds with a pretty high affinity, 5 micromolar. Isoproterenol binds, it's an agonist, so it enhances the effect of epinephrine. Also has a pretty high KD. Propranolol is an antagonist, meaning it inhibits the response. These are all involved in this system as binding sites on the receptor. So how does this work? Okay, so epinephrine binds the receptor in step 1. It's the beta-2 adrenergic receptor. From there, the G protein, shown in green, dissociates from the GPCR. From there, the alpha unit of the G protein displaces GDP, and GTP takes its place. GTP goes to adenyl cyclase. Adenyl cyclase transforms ATP to cyclic AMP through catalyzing the reaction. From there, we get one of two mechanisms. We get cyclic AMP, which activates pKa. pKa phosphorylates, causes phosphorylation. It's a kinase and leads to a cellular response to epinephrine. Or we can have cyclic AMP is degraded and reverses the activation of pKa. So both of those occur. That's kind of how. And then from there, we have all kinds of, and we'll talk about the details of those. We have all kinds of events that are associated with that. This is the first messenger part leading into the second messenger part, which is the cyclic AMP. So activation of few GPCRs lead to the activation of few adenyl cyclase enzymes. Every active adenyl cyclase enzyme makes several cyclic AMP, thus activating several pKa enzymes, again a kinase. These activate thousands of glycogen degrading enzymes in the liver, and in the end, tens of thousands, if not hundreds of thousands, of glucose molecules are released into the bloodstream. Kind of reiterate the same point that we discussed earlier, where one epinephrine molecule, through a series of steps, modular events, can lead to significant tens of thousands, if not hundreds of thousands, of signal amplification. Again, cyclic AMP is a common secondary messenger. A large number of GPCRs mediate their effects using cyclic AMP. Cyclic AMP can activate or inhibit. It can be inhibited, I guess I should say. So GPCRs can either activate or inhibit cyclic AMP. An interesting note, human genome has about 1,000 GPCRs, if not more. They're hormone receptors, growth factor receptors, neurotransmitters, and they're hundreds of different GPCRs that are responsible for very similar processes like taste and smell. And we don't even know many of the ligands. Most of them have cyclic AMP as a secondary messenger. Again, cyclic AMP is able to mediate multiple signals. So for instance, pKa is localized to a particular structure by an anchoring protein. In this example, pKa initiates a phosphorylation event that causes ATP to be cyclized into cyclic AMP. And different anchors are expressed in different cell types to determine how that works. So it can be upregulated, downregulated, many different ways of regulation. Cyclic AMP is very common in GPCRs. Cyclic AMP is a second messenger. There's other second messengers such as inositol 1,4,5-triphosphate or IP3 and or things like calcium. So if we have a hormone receptor example here, we go through the standard steps to step 4, but at step 4, instead of going to cyclic AMP as a second messenger, we're going to go into IP3, where a phospholipase C translocates an IP3. That IP3 leads to the nuclear envelope. That IP3 binds a calcium ion channel that causes the export of calcium from the nucleus. That calcium export of the nucleus, then that calcium as a second messenger will activate protein kinase C on the cell surface, and that protein kinase C produces the main cellular response for the hormone. So secondary molecules are very, very important in these whole processes. Many sensory perceptions mediated by GPCRs. Things like vasopressin and epinephrine, which are hormones. You have light, you have odorants, and you have taste. And what the difference is these modular processes. So in the hormone, we have cyclic AMP that goes to pKa. Some are upregulated, some are downregulated. You'll notice the difference between vasopressin and epinephrine is in vasopressin, downregulation of pKa causes the effect, whereas in epinephrine, the upregulation of pKa is important. Light, we have cyclic GMP downregulated that leads to an ion channel. In odorants, you have IP3 and is upregulated, and cyclic AMP are upregulated, and then we have calcium ions that are both calcium or sodium that are upregulated. In taste, we have cyclic AMP that's upregulated, then we have a pKa effect, and then we have a downregulation of an ion channel. So all of these different things produce, and then we haven't talked about things like increasing gene expression on GPCRs, but this is how GPCRs effectively perform their jobs. The next class is a tyrosine enzyme-linked receptors. The classic example is a tyrosine kinase. This class of molecule is very much a cancer target. So things like VEGF, TRK, EGFR, these are all very, very common. So VEGF is vascular endothelial growth factor. It stimulates angiogenesis. You'll see this. They'll try to downregulate it in cancers, but they're trying to upregulate it in wound repair. So these are the kind of examples that you're going to see. And what you find is you have a binding domain that's exterior. On the inside of the cell, you have a signal. So that's the ligand binding domain. On the inside of the cell, you have a tyrosine kinase activity. Usually, it's an autophosphorylation that is the first messenger that will lead to a second messenger system that will again cause some kind of outcome, gene expression, opening of a pump, etc. Third class, we have guanylyl cyclases. These work by converting GTP to cyclic GMP. Again, works directly, so it binds, it then converts GTP to cyclic GMP. That can be used as a secondary messenger. Sometimes it can also be used, these guanylyl are used as endotoxin receptors, where bacteria figured out that this is an endotoxic response and can cause all kinds of problems. So here's an example of the much promised insulin signaling cascade, where glucose then can be GLUT4 can bring in glucose into the cell. So if you look at the insulin receptor, it's a tyrosine kinase. Insulin binds. We have an alpha and beta. We have an autophosphorylation event that goes to PIP2 pathway that leads to a PIP3 pathway. This leads to multiple other pathways, including GGP conversion to GTP. That GTP then initiates GLUT4, so we get different signaling events off the same cascade. You'll notice that we get cell differentiation off that cascade, we get glycogen synthesis off that cascade, and we also have uptake of glucose all off that same cascade. So there's three different responses off this PIP3 to PKB pathway. All three go signal for the cell to do something. So that's kind of, that's the complexity of the system is, and you can have multiple things off the same, and this some of the modularity we've been talking about. Okay, next class is integrins. Integrins mediate cell adhesion. They interact with what's called the RGD domain, where we have an arginine, glycine, and aspartate residues as a domain. Binding domain, collagen, fibrinogen, fibronectin all bind to that extracellular domain. This can trigger cytoskeletal rearrangements and new gene expression, and then it also can it basically takes the signal from the outside, brings it to the inside, and then we take that signal back outside and cause extracellular response. It triggers it. So basically, collagen or some other binding event occurs on the exterior side. On the interior side, we get a response. We get a change in cell polarity. Survival based on all kinds of things. So if you have damage to the extracellular matrix, changes in cytoskeleton structure, that will lead to gene expression. From the response, talon in the cytoskeleton triggers an inside-out signal. So then that's what happens. So gene expression is commonly, or setting of cell polarity. Those are all common inside signals. That signal is then sent outside, and on the outside we can have a response including increased cell adhesion, migration, more assembly, a building of extracellular matrix. So those are the important parts of what we're talking about. If you look in the center, there's an alpha and a beta integrin. Alpha is in pink. An alpha integrin has several binding sites for divalent cations. Beta has extensive cysteine residues that has extensive disulfide bonding, which is important as a receptor. And these are very, very that stabilizes that protein, the cysteines do. But these allow for collagen binding, heparin binding. They can also tell damage to the exterior side of things. Again, we can get all kinds of inside-out signaling that occurs with integrins. Cell damage can be identified with integrins, as well as high polarity environments, again with the alpha subunit. Gated ion channels are the next extracellular biosignal that we'll talk about. These regulate transport of ions across the cell membrane. They can respond to changes in membrane potential or ligand binding. They're very important in the nervous system, as we talked about earlier, for things like GABA, ionotropic glutamate, nicotinic acetylcholine receptor, which we'll talk about, and voltage-gated sodium channels. Very briefly, we've already talked about gated ion channels a little bit, but this gives an example of what you would see. So you have voltage gated. If the potential changes, we're going to start using ATP, and we're going to convert that to ADP to pump out 3 sodium channels and bring in 2 potassium. That'll change the potential of the cell. We can also have calcium, chlorine, potassium, all these ions, and we can move them up or down their electrochemical gradients. Again, these are important in nerve signaling. You can see we have a depolarization event where these pumps work. They pump in the cell, and to repolarize, we're going to use voltage-gated potassium channels. That signals in step 3 the acetylcholine secretion in vesicles. So acetylcholine vesicles are secreted across the synapse, where we have acetylcholine receptors. Those acetylcholine receptors again cause an action potential to occur in the neurons. That's what initiates the nerve response in a cell. These voltage-gated sodium ion channels are very, very cool. So if you look at this figure in 4, where the voltage sensor is, what you basically have is a voltage sensor. When voltage changes, the tether, which is shown in 1, will open or close based on the voltage that it's presented with. And that will allow ions to come in and out of the cell. Usually they're going to go down a chemical gradient, obviously, right? Because it's a channel, not a pump. So these, if the voltage differentiates too much, you can just open up this, it'll cause that voltage sensor to trigger, and that tether, which blocks the channel, will conformationally change the protein and will open up and allow sodium or other ions to flow. This one's specific to sodium. There are other voltage-gated ion channels that will are specific for their specific ion, as we've discussed. Okay, last we have direct regulation by hormones. So what we have is a hormone that comes in and binds to its receptor. This usually occurs in the cytoplasm. Up to this point, the hormones run around in the blood bound to serum proteins. Once a hormone crosses into the cytoplasm, it loses that, it binds to the receptor. In step 2, you see it causes a conformational change in the receptor when the hormone binds, and it forms homo or heterodimers with other hormone receptor complexes, and that allows it to become a transcription factor. That transcription factor binds to the complex for transcription, and that causes gene expression. That's how a hormone works. And the hormones can be responsive to numerous different things. Big picture. We have different cellular components that are present in all different kinds of organisms, from mammals and plants and bacteria. Mammals have broad diversities of these. Plants do as well, and bacteria use a subset of what we have available to us. You can see that there's all different kinds of these signaling components that exist naturally. People usually spend their entire careers looking at one thing. Let's dive back into acetylcholine receptors. Acetylcholine receptors mediate. They can increase the blood. They can increase the pumping action. They open up channels, the blood vessels. So how does this work? So if you'll notice that an alpha helices, each subunit has 4 alpha helices. That's what's shown on the left. Each alpha helices M2 domain is important on the inside of the channel. Each subunit, okay, has a specific role. There's 5 of them that are put together to form this acetylcholine receptor. Acetylcholine receptors bind, you can see right next to the alpha subunits, and on one, and the alpha and beta in the other. Once they bind, they open up that channel, and it opens the for the influx of sodium and calcium. That's how an acetylcholine receptor works. This kind of gives you a little bit top, so this is looking at it from the top view. You can see what's closed normally. Once acetylcholine binds, it causes a conformational change in the protein that opens up the channel, and that allows the calcium and other ions to go through. It's a very simple binding mechanism. This example, if you look this up as a molecule of the month, and I would encourage you to look at it, it also provides, again, angiotensin II receptors. When we talk about this, I don't want just to come across as we're just telling you this biochemistry for the sake of telling this biochemistry. It has a function, and when we talk, heart disease is a huge problem. Congestive heart failure is a huge problem. Understanding why and how these treatments solve these problems biochemically is very, very important. It's an important tool in your toolbox. We're not just up here trying to give you the biochemistry. We're trying to give you a frame of reference for everything that we do, so that you can actually use it and apply it in your profession as a tool. That's what this is. And if you get in, you're trying to figure out, you're trying to explain to a patient, you're just trying to figure out if a drug is safe. You're trying to assess, safety information, side effects. All these kind of things occur at the biochemistry level, and that's why we're kind of pounding in all these things over and over and over to give you the ideas of how to address problems in your, when you face them in your career. All right, that should be the lectures for the week. We went relatively fast. The advantage of video is, and I would encourage you to do this, if you don't understand something, slow down, go back,